Hi everyone, so today we'll be going over Pearson Edexcel 2024 GCSE Paper 1. This is a predicted paper and we'll go over all the questions and all the topics that are likely to come up next on Friday. However, make sure that you still cover every single topic, and if you want this paper you can go to my pinned video, there's a link to get it, or you can go down on the comments below. So let's start. Question 1. As we grow, we make new cells by mitosis and meiosis. The cells can be specialized. So figure 13 shows a diagram of a sperm cell. Describe 2 ways in which the sperm cell is specialized. Specialized means that they have special features that not every different type of cell has. So as you can see here, it has a tail, and this is one of the features. Not every cell, like a red blood cell, does not have a tail. And another feature is that it has many mitochondria. Some other features that you could have are that it also has a sac with enzymes. It's that it's streamlined, meaning it's very thin and it's long, which is unnatural since most cells are actually just more like a circle. And also that it has 23 chromosomes. Complete the table to show the results when a cell divides by mitosis or meiosis in humans. So human body cells, except gametes, have 23 pairs of chromosomes. So number of daughter cells produced in mitosis, this is 2. This is because they only divide once, while meiosis divide twice, so it is 4 daughter cells produced. So number of chromosomes in each daughter cell in mitosis, this is 46. This is because it replicates and divides once, so it's 46. Or it will be 23 pairs, while meiosis is just 23, and it's just chromosomes, not in pairs. Question 2. So figure 4 shows 3 cells. What is the structure of X? So we see X. So it talks about these, that over there. You can see that they did not point to animal cell. So this should already show us that it's something that only a plant cell and bacterial cell has. So it is a cell wall, that structure over there. While an animal cell does not, it only has a cell membrane. So the bacteria cell in figure 4 shows a flagellum. What's the function of it? So this is over here is it. So as you can see, it's kind of like a tail, so it allows movement and mobility. It allows it to basically swim and go in a direction that it wants to. So give one other differences between a bacterial cell and an animal cell. One difference is that a bacteria has no nucleus, and also it can be that it has a chromosomal DNA, and bacterial cell has a cell wall, as you can see here, while an animal cell does not. If you can see here, bacterial cell has a loop of DNA, while an animal cell has a nucleus, which is shown here. Question 3. Proteins are made up of amino acids. The table show DNA bases that code for some amino acids bound in protein. So part of the DNA coding for protein is this. So state the order of amino acids by the sequence of DNA. So we know that it goes by 3. So amino acids, they code by 3. So 3 DNAs makes one amino acid. So if we go, we can see that TAC, CAA, TTC. So we know that there's 3 amino acids. So we just need to match these to the box. So we see TAC, first one is met. Then we see CAA, next one is val. We see TTC, which the last one is this. That's just how you do it. These amino acids will be joined together by protein synthesis. Describe during which stage of protein synthesis will these take place. This is during translation. This is when it will be joined together to make the chain of amino acid, therefore the protein. Complete the sequence by putting a cross X in the box next to your answer. So amino acids are joined together. This is at the ribosomes. This is where translation occurs, and this is where they come together and they join together. In the nucleus, this is where transcription occurs, which is where the sequence of amino acids and mRNA goes to the ribosomes. So B, DNA can code for amino acids in active site of enzyme. Explain the role of active site in an enzyme. So this question is always the same. Whenever you see enzyme, I recommend that you write these 3 things out each time because you're guaranteed to get at least 1 mark, no matter if you know the answer or not. So active site of an enzyme has a specific shape. So this is kind of how an enzyme looks like. They're always going to have an active site. So this always has a specific shape that is only complementary to the substrate that it wants. So it can only usually go to one substrate. So we have a lot of different enzymes for a lot of different types of substrate. So it binds to the substrate to form an enzyme and substrate complex. This is a very big key word that you usually need. And therefore it is catalyzed. So that allows the reaction to take place. And what the reaction is that it either joins together two substrates or it breaks down that substrate, whatever the reaction is. And you can also refer to the lock and key mechanism. C. Mutations can occur in DNA. Describe what effect a mutation could have on the action of enzyme. This again is another very common question, and it's always the same 5 that comes up. So this could be a 5 marker. So every bullet point is a different mark that you get on the mark scheme. So you only need 3 of these, but I recommend you learn all of this and you always put down the exact same thing every time you see a mutation, regardless of what the question asks, just even if you don't know, to gain as many marks as possible. So a mutation is a change in gene-base pairs, and this causes a change in the amino acid and the order of the amino acid. So because it changes the order of the amino acid, the shape is going to change, so it might become ragged like that. So this prevents it from binding to substrate. So the substrate is like this. Clearly the substrate isn't going to be able to fit into that. This means that the enzyme can no longer function. They can no longer catalyze the reaction, so it cannot join it or join two together anymore. So enzymes could be more effective in a different way. So D, describe how named factors affect the rate of enzyme catalyzed reactions. So there is 3 things that affect the rate of enzyme catalyzed reaction. This is temperature, pH, and concentration of enzyme and substrate. I recommend that you remember every single one of these because they might not ask it in a 6 marker. This is a really lovely question. They might ask it as a 2 marker, a 3 marker, a 4 marker. So just always remember the points and how that affects it. So we'll start off with temperature. So if the temperature is too low, it just means that there isn't enough energy to make the reaction actually occurs. There's not much energy for the enzyme to actually meet the substrate. So for humans, the optimum temperature is 37 Celsius. But once it goes over 37 Celsius, it becomes too hot for them. So the enzyme changes shape. So therefore, it means rate of reaction decreases, and that means the enzyme is denatured. What enzyme denatured mean? It just means that the enzyme changes its shape. So pH, the optimum pH for most enzymes are around 7.3, so it's around 6 to 8. So either side of the optimum, this is kind of like how the graph would look like. So either side, it will change the shape of the enzyme just because this makes it denatured, just because it's not their optimum pH. So then again, the rate of reaction decreases because it's less likely for an enzyme and substrate complex to occur. So substrate and enzyme concentration. Higher concentration equals faster reaction, and this is due to more collisions. So they're more likely to collide to each other since they're more in a certain amount of space. So because it's more likely to collide to each other, there is more collisions, meaning more rate of reaction. And this is until the maximum rate has been reached, because there's only a limited amount that you can get. This is the exact same in chemistry. It's the exact same in physics. So it's worthwhile to learn these. Question 4. Arthritis is a condition that occurs when cells in joints get damaged or destroyed. Stem cell therapy can be used to treat arthritis. Discuss the benefit and risk of using stem cell therapy to treat arthritis. So because it says 4, we can split it up so it's 2 benefits and 2 risks. So always take how many marks it is as an indication of how many points you need to write down. So one benefit is that stem cells can differentiate. So this means that it can become specialized to a certain type of cell. This means it can replace any damaged cell, no matter what type of damaged cell it is. It could be a red blood cell, it could be a skin cell. So then this also reduces the symptoms of arthritis since it's able to replace all those damaged cells. What are the risks? The risk is that some of the new cells might not function correctly. It could be that stem cells continue to divide, and what we know is that the more cell division that occurs, the more likely there could be a mistake in that cell division, so that can cause things like cancer, since that's how cancer is caused, is by not stopping and keep cell dividing, and a mutation happens within that cell division process. And also risk of side effects such as them rejecting the cells, and symptoms may also be worsened. So most likely if they ever do go on the stem cell therapy, they need to get even extra pills such as rejection and immune suppressor pills. So that might not be a good thing, not might be a comfortable thing for them to go through. So 3I, draw one straight line from each stage of the cell to its description. So these are the stages of the cell, these are description. So interphase is the first stage of cell cycle. This is where the DNA is copied and doubled. So it is over here. And then we go through PMAT, where this is prophase, metaphase, anaphase, telophase. These are the next couple of the cell cycle of what happens. And then cytokinesis is the last one. This goes here and interphase goes here. Cytokinesis, this is when the cell divides in two, and it's the last stage since 2 comes. So if we go through here, this is PMAT, metaphase, and anaphase. So question 5, figure 17 shows light rays entering the eye of a person with a normal vision. So describe how the light rays are focused to give a normal vision. So as you can see over here, the light rays are going in a straight line. However, once it reaches this lens, which is this part over here, it bends in and it refracts. So that is the first thing that we say. We say light rays bends in by the lens. And what they do is that it basically bends so that it can focus on this point, which is the retina, so that we're able to see. So if you ever see that there's a diagram, there's usually a diagram there to help you. So just try and describe the diagram if you're unsure what the question is asking you to do. So 2I, figure 18 shows light rays entering the eye of a person with an eye defect and 2 lenses that can be used to correct the defects, X and Y. So explain which lens would correctly the eye defect. So you can see here that it just focuses too early. That is what the problem is. So we can see that lens X will be the one that actually fixes it, just because it's a diverging lens, or another word is a concave lens. It's concave, kind of, it goes inwards. That's why it's kind of like a cave. So this means that it will spread out the light rays. Because it goes too early, it will just spread it out and delay it so that it actually reaches in the correct time. The Y would just make it come closer. So this would be if the defect was that it would go too late. Question 6, neurons form part of the nervous system. Name structures A and B. So if we see here, this is how a neuron looks like. You need to remember that, since they might ask you to identify what type of cell this is. So we can see structure A over here. These are the dendrites or dendrons. And this over here, you can see whatever is circle. This is just the nucleus. Complete the sentence by putting a cross in the box next to your answer. The axon carries information along the neurons. This is as electrical impulses. So basically there's this neuron. There's the neuron. It goes down, this is by electrical impulses, and it goes down to the next one. So while within the electron, this is electric. However, in between these gaps over here that happens, this is where the chemical messages actually go, and then it gets converted back at the top to electrical and it goes down. So that's why some people may get confused. So describe the role of the myelin sheath surrounding the axons. So this over here is the myelin sheath. So what this does is basically, it's kind of like a blanket on top of it. It insulates the electrical impulses, and this allows it quicker conductance. Basically speeds up the transmission, just sends it a lot faster, since it kind of just jumps between it. It's so much easier than just going through it really, really fast, because the electrical impulses can only travel at these points over here where there is no. So it's able to go really fast instead of just going through the whole thing. Oh, I missed. And then there is a gap between neurons. State the gaps between these neurons. These This is what I was talking about, these gaps here. These are called synapses. So humans have reflexes. Describe the route of an impulse through the reflex arc. So I'll describe this using a diagram. So if you see over here, we'll take this as a hand. This is kind of like when you catch something really hot and you kind of put your hand out as soon as like you feel it, and that's just something that automatically happens. You do not have to think about. So the brain is not involved in this process. So the brain is not involved. So basically your hand feels it. So there is sensory neurons in your hands. This basically sends an impulse to your spinal cord, which is here. And then from the spinal cord has a relay neuron. And this relay neuron just basically, its function is just to transmit what the signal is saying from one neuron to another, kind of like a relay. And this relay neuron, it will go to whatever the effector is, and it goes through that. It will send its message to the motor neuron. And in this case, what we want the response is for your hands to take away. This will happen through muscles. So, and then it will go to the muscle, and it will send the effect to the muscle, so that initiates whatever response. However, this can also be something like an effector such as a gland, if it wants to secrete hormones, like let's say adrenaline hormones, if you just want to have like your flight or fight response. So if you go through the answer, it detects the stimulus from the environment, and then the sensory neuron passes to the spinal cord, and then relay neuron to these is inside the spinal cord. Then relay neuron passes on to the motor neuron, and then motor neuron passes to the muscle. Not the best drawing. And then muscles initiates the response. It's whatever the desired response is. So this can actually be a 6 marker. There's 6 different points. So do be prepared that 6 marker. And if they ever give you like a diagram or something, make sure you always relate back to that diagram, since you will get some marks on application. Question 7. Hemophilia is a recessive sex-linked disorder, and this family prodigy shows the inheritance of hemophilia. So before you look at any questions, let's make sure we understand what the diagram is even telling us. So we can see boxes are male, and if there is dark, it is a effective male, and this is a carrier female. Okay, so let's see. State the sex chromosomes of B. So we can see B. So we can see over here it is a circle, so it's female, and we know the female is XX. If it's a male, it will be XY. So explain why the male offspring from A and B do not have hemophilia. So if you look here, these are all the unaffected males. So we can see that they do not inherit the allele. We'll state the obvious in the beginning. They do not inherit the hemophilia allele. They do not have it. So, and this is, as we can see that the effective male has with its X, and then the carrier is XX. However, it's never complete, like there's never a full circle. So we can already detect that it is the X that carries this allele of hemophilia. That's why it's so much more common in male, as you can see, because they only require one X, and this X, male always get their X from the mother. So we can see that male receives X from the mother, and we can see over here that the male, which is B, you cannot say that they're unaffected, because unaffected won't give you the marks because they could be a carrier, but we can see over here that they do not have it, and they are not a carrier. So we need to make that very specific with what we're saying. So you can see, because B is homozygous dominant, you could say it in that sense where it's a bit more biological. So using the Punnett square, calculate the probability of individuals C and D to have a child with hemophilia. So let's see what they are. So we can see this is a unaffected male, so XY, and this is a carrier female, so XX. So if you go over here, we'll say that H is without, and H is without. Y we do not consider, and H is if they do have it. So we'll put it on each one. So we'll put X, H, and Y, and then we'll have XH here. We'll have X just normal H. And then basically what we do is we basically cross multiply and see what the different types that can come up. So XH XH. You can see here XHY, XH with XH, and we can have XH with Y. You could also just do HH, HY. So whatever is easier for you so that you can understand, but make sure it's very clear because the examiner is marking this. So as you can see here, you can kind of see this is not going to get it, not going to get it, not going to get it. So none of them are going to get it. However, this over here is going to get it because they only have one X and this one X is. So if you see, this is 1 over 1, 2, 3, 4. So there is 1 in 4 probability of the child having hemophilia. So that's how you do it. So don't overcomplicate it, just go very simple with it. B, hemophilia can be treated using a blood clotting factor produced in a fermenter. So the conditions of inside the fermenter have to be carefully controlled. Explained one named condition which must be controlled in the fermenter. So we see it's 2 marks, so we need to make 1 point and 1 explanation, even though there is multiple ones, and I'll go through each one of them now. So the one I chose, the one that I think everyone is a very common logical thing to say is we need to make it aseptic condition, so we make sure that it's completely sterilized because this will prevent the growth of unwanted organism. Unwanted over here is a key word, you must have it. And also because it prevents contamination, so we know that it's complete to be treated and it won't be infected by something different. What also you can have temperature or pH. This is because it provides optimal conditions for growth and it prevents enzymes from denaturing. You can also have nutrient levels, and this is because it provides optimal conditions for growth. And you can also have oxygen, since this allows aerobic respiration. So it provides, again, optimal conditions for growth and prevents anaerobic respiration, which is what we don't want. So question 8. When bacteria divide, they replicate their genome and synthesize their cell wall. So figure 12 outlines the stage of bacterial replication, as you can see over here. So A, penicillin inhibits the synthesis of a cell wall in bacteria. This is a very important fact that we need to learn, since we're most likely going to need to link that back into the question. So explain the effect of penicillin on bacterial and human cells. So make sure that we mention both of them. So let's see. Penicillin prevents the bacteria from dividing as they cannot make a cell wall. So as you can see here, it inhibits the synthesis. Synthesis just means make. So it prevents the bacteria from dividing because they cannot make the new cell wall, and it is crucial for their functions. However, as we know, every single type of human cell do not have a cell wall. So no cell in the human has a cell wall. So this means we're just unaffected by penicillin, since the only thing they do is inhibit cell wall, meaning that our cells can continue dividing as normal. So B, penicillin is isolated from a fungus and was the first antibiotic used to treat bacterial infection and still widely used today. Scientists have genetically engineered bacteria to produce large amount of penicillin. Describe how the scientists would produce a genetically modified bacteria that produces penicillin. So this is just genetic engineering. We'll go through this really. So what happens is that we can see over here, this is the loop of DNA, the plasmid. We use the plasmid of a bacteria since they do not have a nucleus. And we take a restriction enzyme, this is what's called, and this function is basically to cut out the gene that we want. So if this is the gene that we want, we take this enzymes, and the enzymes know exactly, because they read the code of the gene, and they know that we want to cut out this part. So then we use ligase to join the DNA molecules together. So we take it out, remove it, and we try and put it into the DNA that we wanted to put it. And ligase is basically a glue. It is also an enzyme, and its function is a glue. So then we cut the gene from the genome of the fungus. As we can see, it's isolated from fungus. Make sure that you always relate back to what the scenario gives you. That's how you get the marks. And extract a plasmid from the bacteria. So then what we do is that once we get the plasmid that we want with all the correct DNA, we put it back into a bacteria. And because we know bacteria, they reproduce exponentially, which means that they increase really, really fast, so that out of in like 1 minute, we can get so much bacteria, which is so much faster than human cells or any other type of form of division with cells. We put it back into a bacteria, and that means that they can produce a large amount, since they'll start replicating the plasmid that we want, and we're able to extract this and use it for, we can use it so that we can treat bacterial infections. Okay, so C. MRSA is a bacterium that has evolved to become resistant to antibiotics. With reference to Darwin's theory of evolution by natural selection, explain how MRSA bacteria have evolved to become resistant to antibiotics. So we need to make sure that we link it back to Darwin's theory of evolution by natural selection. So as soon as you see this, we kind of already know that our structure of our answer is going to be in terms of the common answer for natural selection. That is basically what we're going to go around. So as soon as you see natural selection or evolution, these are the same 7 points that we're always going to use. So it's worth learning them. And then whenever you see it, you can just add it. Of course, changing the things, the subject we're talking about. So first of all, we can say that bacteria reproduces rapidly, generating a large population. But then with every single type, there is mutations that occur. So this causes a variation among the bacterial population. So, and what this variation is that it develops a resistance to antibiotics. And this is through a mutation that occurs. This is just because it's normal with any type of population that is growing at a large rate. So then the antibiotic treatment exerts a selection pressure. So what does selection pressure mean? It just means that it will kill off anyone that doesn't have antibiotic resistant, which means that the only thing that are alive now are antibiotic resistant bacteria. And because they're alive, no matter how much we give them, they're always going to stay there. So this means that they're going to survive, meaning that because they survive, they're able to reproduce and pass on the gene, meaning this gene is inherited. So non-resistant bacteria do not survive, so they do not pass on their gene. So the only thing that is left is the antibiotic resistance bacteria. So that is how it develops. So the level of antibiotic resistance in a population of bacteria increases. So question 9. Charles Darwin studied a variety of finches on the Galapagos Islands. So he used the information to develop his theory of evolution. Some of the finches are shown in the diagram, which is shown over here. This is a diagram which just shows what the ancestors are and how they evolved from one another. So like, how are they related? So we know these are all related to the same ones. However, this is not related to this one. The only way they're related is through this over here. So the lines kind of connect everyone. So state the genus of this and the species of the large cactus around the finch. So if you see over here, we can see that the genus is over here. This is whatever is the second thing after the common. So the genus is Geospiza, and the species are this, is whatever there is once, since that's like specific to them. So there's actually a C over here, but it's not shown. So it's Construs. So 2I, suggest how the size and shape of beak enabled all these types of finches to survive. So how they do is that different beak size enable them to have different finishes to feed on different types of food. So this happened due to a mutation, and because they have different ones, so ones with smaller beak can get nuts, however ones with bigger ones can have fish, and this means that they started to have different food categories, which means that there's less competitions between species. So this is always the main point that we would like to go back to. So always try to go back and see how does something create less competition between species, and this is how you relate it back. That's how you get the 2 marks. So complete the sequences by putting a cross X in the box next to your answers. Darwin finches are an example of speciation, and this is due to geographical isolation. So if you see here, These are all the variety of finches in one island. So this means that there's some places that they're not allowed to see from each other, and because they're all in one island, they all have the same type of environment, so it's geographic isolation. So B, suggest how these species of finches could have evolved. So again, we see evolved, we try to go back to the same structure that we know for natural selection and for evolution. But when we go back to natural selection, however with evolved, we start talking about survival of fittest, natural selection. So the variation, you can see that you will tell that it's the same answer every single time, just different ones. So variation between species of beak sizes and shape, and this is due to a mutation that happens. So this means that there is competition of resources, so there is survival of the fittest, so whoever is best adapted to those environments can survive, and because they survive, they're able to pass their genes. However, those that are not as well, they won't be able to reproduce. That means that they're not able to pass on their genes, so their genes kind of gets taken away, which is just natural selection. As you can see over here, just mentioning it gets you 1 mark. So question 10. In 2012, there was an outbreak of whooping cough in the UK. The graph shows the number of cases in the whooping cough in UK from April to December. So let's make sense of this graph. This is the number. Sometimes they might put like thousands in brackets, so make sure that you do remember that, and you remember it when it comes to your calculation. However, this time they do not want to trick us. And over here is the months. So describe the trend shown in the graph from April to December. So we can see over here that there is a rise. So there is a rise and then there's a decrease. And this changes in October. While there is a decrease here in June, we just want the overall trend. So we would take this as an anomaly. So all of this, it increases, then decreases, and that's just the answer. It's just a describe. So if it does not say explain, we don't need to explain exactly why this is happening. We just need to say what we can see. So an increase in cases until October, then a decrease in cases till December. Okay, in September 2011, there was 168 cases of whooping cough in the UK. Calculate the difference in the number of cases in whooping in September 11 and September 12. So we just need to see this September 2012. We just need to read from the graph. So we can see over here, this is around 1320 if we take a scale and see how much. And all we do is just a simple equation of just minusing them together. So there is a difference of 1,152 cases. Make sure you always show all your working outs, and if you do any reading from the graph, make sure you make your line and how you read it very clearly, since that's your working out marks. So B, whooping cough is caused by bacterium, which grows rapidly in the body. So state the term to describe the rapid growth of a bacterial population. This is called exponential growth. This is just something that is unique to bacteria, so make sure that you learn it. So C, children in the UK can be immunized by the whooping cough. Suggest why outbreaks of whooping cough still occur in the UK. This could be lots of different reasons. So it does say over here 2, which means we only need a minimum of 2 points. They'll only mark us that. However, I recommend putting as much points as you can think of, since it doesn't really say here how many exactly you need. So one might not be correct, at least you have a different one that could be correct. And they'll just mark all the correct ones, they'll just ignore the wrong ones, since it's not negatively marked. So one could be that not everyone has been immunized, there's no herd immunity. Another one is that immigration introduced people who are not immunized. So immunization is not fully effective, just a simple one. And immunity does decrease with age. So describe the response of the human body to immunization. So this is basically saying how are we immune to certain diseases, and this kind of comes from vaccinations, or it comes from being exposed to a certain thing a lot of different times. So we can just say that. We can say it introduces of an antigen. You can say this is through having the virus yourself, or this could be through a vaccination. Kind of always link back, kind of like how COVID, we all we all got the vaccination. So this causes an immune response, and what the immune response is that B lymphocytes, these lymphocytes which are they produce antibodies, and these antibodies, they go against it, and they basically conquer antigens, and they eradicate them. And this means, but not all B lymphocytes does that. There are some antibodies which turn into memory lymphocytes, and they basically take down every single thing that you needed and every single type of reaction that was able to kill these antigens, so that if you do ever get that antigen again, it's a much faster response and it's a much more long-lasting response, so you don't get as much symptoms as you did the first time. Question 11. One cause of color blindness is a change in DNA sequence. This results in a production of different proteins in cone cells and retina of the eye. Explain how a change in DNA sequence of a gene can result in a production of different protein. So again, it's just talking about mutation and a change of DNA sequence. It's the exact same as what we were doing from before, the same structure. So changes of the sequence of the mRNA. And the mRNA, the sequence of mRNA, this is produced in transcription. So this leads to a different amino acid. This is because it's the sequence of mRNA that determines what the amino acid is. As you can see, there are 3 different ones that creates the amino acid. So if you kind of look back here, just to make a bit more sense, you can see, let's say, if over here, if one of the amino acids changed and this changed into a T instead of an A, and that mutation occurred, this won't be met anymore. This would actually be the less amino acid, and because of that, that's why it will change. And now this whole thing is going to change, and the whole amino acid structure is going to change because that amino acid completely changed, just even though only one of the DNA structure changed. So that causes a different shape and different function because of a shape, because we know that active sites are very specific, and just a change in the active site means that they're no longer able to bind to the substrate, so enzyme substrate complexes aren't able to happen again. So function of the protein changes. Question 12. Strawberry plants grows runners, and new strawberry plants develop along the runners. So the new plants are genetically identical to the parent plant. This already tells us that this is either through asexual reproduction, which is through mitosis. So this is the parent plant, and these are the runners over here. So now name the cell division that results in the plants of the new. These new plants, we already made that. That is mitosis. This is because mitosis, it creates genetically identical, whereas meiosis creates non-genetically identical daughter cells. So farmers cut the runners and sells the new plants. So suggest the advantage of producing the new plants in this way. As you can see, it's 2, so we only really need 2 points. However, I recommend doing as much as possible just so you can maximize the amount of marks you can get. So what it is, because they are genetically identical, it could be that the same characteristics of the offspring as the parents. So this means that you can take whatever the best characteristics of 2 parents are, and you can keep reproducing those ones. So you clone them so that they're identical and they always have the best characteristics. This also can be, it's easier to generate new plants since it's also way quicker to produce them. And it's also very cheap because the plants will not run out and there's no need to buy new plants and new seeds. If you ever say something's cheap, make sure you explain why, since just saying cheap or just saying expensive never give you the mark unless you really have to elaborate on it. So some students extracted DNA from these strawberries, and this is the diagram shown. So stage 1, grind the strawberries in soapy water. Stage 2, filter the mixture into a boiling tube, and this is the strawberry filtrate. Stage 3, slowly produce ice called ethanol in the strawberry filtrate. Stage 4, remove the DNA with a wire loop. So suggest the purpose of stage 1 and 3. Stage 1, this is grinding the strawberry. This is just to break open the cells so that you separate the components and basically releases the DNA and dissolves protein. And stage 3 over here, this is so to precipitate the DNA from the solution and basically just being able to separate it from all the other things. This is what the cold ethanol does. So a section of a DNA from a strawberry is shown in the diagram. How many codons are shown in this section of DNA, and put a cross next to the answer. So we know a codon is every 3 DNA. And even though it does show 2, we don't need to make them separate because this is just how a DNA is. In real life, when they go for transcription, we only use one of them as a template. So if we just go of every 3, we can see that there is 1, 2, 3, 4. So it's 4. So this DNA is found in the structure within a cell of a strawberry plant. On the diagram of the plant, draw the name of the structure containing DNA. So what it is, we can just put here. This is the permanent vacuole, so we can't actually draw here. We'll draw in the cytoplasm, which is in this area, and because this what contains DNA, we know that this is the nucleus. And it says label, so make sure that we label it. So question 13, figure 10 shows the number of people diagnosed with sexually transmitted infections, STI in UK during 2017. State the sexually transmission affected has a medium number of people diagnosed. So how we find medium is that we take it from top to bottom, and then we just take the middle one. So we'll just take it from lowest to most, 0.1, 0.6, 0.8, 1.1, and 3.7. So then we take 1 from each side. The middle is 0.8. So they want to state the name of it, so this would be that over there. The population of UK in 2017 was 66 million people. Calculate the total number of people diagnosed with chlamydia in UK in 2017. So how we do this is that we do 66 million. Let's convert that so that it's in numbers. So 1, 2, 3, 1, 2, 3. And we will just divide this now by, because this is number of people diagnosed per thousand, so we need to make sure our number is the same so that we can actually use it on the table. They always use that to trick you out, so make sure you read what units they want it in. So divide this by 1,000. This will be 66,000. So this is chlamydia, you can see over here. So it's per 1,000, it's 3.7, so we'll just times this by 3.7, so over here it's 244,200. 244,200 people. Okay, so state why chlamydia can be described as a communicable disease. A communicable disease just means that it can be passed from person to person. Another reason it could also be that it's just caused by bacteria. And that's the definition of a communicable disease. So give one way transmission of chlamydia can be prevented. This is to just avoid sexual contact, since we can see over here it clearly says it's sexually transmitted. You can also screen people for infection, or you can treat the infection and give antibiotics, and that can also be prevented that way. So explain why chlamydia can be treated with antibiotics. They can be treated since they are just killed by the antibiotics, and this is because chlamydia is caused by bacteria, so antibiotics only work with bacteria. And that's the end of it. If you have any questions, please let me know and I'll go back to all of the questions. If you want, this will later be uploaded onto our YouTube if you ever want to go over it. If you want the paper itself, it will be in the comments, or if you go to one of my recent videos, there is a link in one of the comments over there. And please follow our TikTok. We have questions every single day to help you with your exam. Thank you everyone for joining.